This presentation covers materials that are in Chapter 4 of your textbook for Microbes and Society, Bio 118. The first part, over the history and understanding of genetics, I will follow very closely with your textbook. I think that there are some parts that are important, though, in understanding the way that DNA, RNA, and protein interact. That is, that DNA is responsible for the manufacture of protein. In looking through this chapter, you should be able to talk a little bit about the research of Mendel and Morgan, and be aware of some other scientists that have been involved in the last 150 years who laid the foundation for recognizing DNA as the hereditary substance in microbes and other living things. You need to explain how the sequence of bases in DNA molecule serve as a genetic code and how that code ultimately gets transcribed to messenger RNA and then translated to a protein. I will speak briefly at the end of this about some applications resulting from the sequencing of genomes of microbes and humans and some of the possible applications and consequences of that. When we're talking about heredity, we are talking about genes, DNA, and when we look into the future at possibly manipulating DNA to get a more desired result. When we first start talking about this research, we do have to talk about Gregor Mendel, who in the 1860s started working with pea plants. He found that there were 7 inherited traits that he could identify and work with. And he came to the conclusion in his research that there was some factor that was transmitted from each of the plant parents to the child, and that they occurred in pairs. That is to say that a parent cell or a parent pea plant might have 2 factors associated with a characteristic, and it would pass one on to its child that the other parent also had 2 factors that might pass one on to the child, and that one of these might dominate, or there may be some that were codominant or some that were recessive. Now, this sort of gives us a concept of chromosomes. He called them factors, but it wasn't until some scientists about 40 years later were doing some research that they were able to attribute tentatively that the factors that he was talking about were located on strands of genetic material located in the nucleus, and they called them chromosomes. It wasn't until Thomas Hunt Morgan did some research around 1910 on fruit flies that you could definitely assign a characteristic that you could see to a certain chromosome. Thomas Hunt Morgan, by the way, was at the University of Kentucky for a couple of years, and the biological sciences building there is named for him. Anyway, he was able to definitely attribute a fly's sex and eye color to certain strands of genetic material. A little bit later on, someone named Willard Johansen was also looking at genetic material and found that the genetic material could really be broken down into certain discrete areas that coded for one particular characteristic or one particular protein, and we ended up with the concept of the gene. So a chromosome carries many genes, and ultimately the factors that Gregor Mendel was talking about became known as genes. The structure of DNA. and there were several people who worked on that beginning in the 1920s. Um, this Phoebus Levine came to understand that there were two forms of genetic material, that there was DNA and RNA. That what we had was uh, deoxyribonucleic acid, which contained a sugar of deoxyribose, and we had ribonucleic acid, which contained the sugar ribose. He also discovered that there were 5 nucleotides. That is adenine, thymine, guanine, cytosine, and uracil. Now, why we have 5, we'll talk about in a few minutes. Erwin Chargaff discovered that there were equal amounts of thymine and adenine that would suggest that somehow they were paired. Additionally, same amounts of guanine and cytosine, also suggesting that they were paired. From that information, along with work by Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick, they are able to discern ultimately that the structure of the DNA was, in fact, a double helix. Watson and Crick are the ones that get the greatest credit for this. Rosalind Franklin was in a lab actually across town. They went to visit her one day and saw some of the work she was doing, came back, um, combined it with some of their own thoughts, and were able to elucidate this shape. They used X-ray crystallography to help them do this. That is, that they shined X-rays or fired X-rays through crystals of DNA, and the scatter pattern that came out suggested the actual structure of the DNA. And what they ended up with was this double helical structure with adenine on one side, thymine on the other, guanine on one side, and cytosine on the other. So each strand of the helix held what would be called a complementary base. And ultimately, what we have is complementary base pairing, which makes replication of DNA very easy. So we've given you a little bit about the history of the discovery of DNA and its structure and function. So let's talk now about how DNA and RNA are involved in protein production. Proteins are a significant part of our body's organic matter. It's the same thing with microorganisms. That is about half of the organic matter in any living organisms, and it provides construction materials for tissues, for individual cells that play a vital role in cell functions. In individual cells, they act as enzymes. In larger organisms, they can act as hormones and antibodies. They also have structural and functional value within individual cells as gateways and for materials to move in and out of the cell, for instance. Proteins are made of amino acids. You really don't need to know the exact structure of an amino acid, but it does have a nitrogen group. And basically, what proteins can do is they can be formed into a couple of different types of structures. There are fibrous proteins, structural proteins, that appear both in body structures and in cells. Examples for us would include collagen and keratin. These proteins are pretty stable. And then there are globular proteins, which are functional in nature. These function, again, in bodies as antibodies, but on a cellular level, they can function as enzymes, they can function as cellular pumps, they can function as part of any of those organelles that we mentioned. The problem with globular proteins is that they are fairly unstable, and they have to be in a pretty safe, consistent environment, or they can be denatured. By denatured, that just means that they can be unfolded in a way that leaves them non-functional. Example of denaturing: if you've ever cooked an egg, when you crack that egg open, it's runny, It's clear, but as you apply heat, it starts to change color and to thicken and solidify. That's because the proteins within that egg are unwinding. Now, proteins have multiple layers of structure. There is a primary structure, which is that amino acid sequence, which is controlled. This is a good place to stop before we move on to our next topic, which has to do with the role of DNA and RNA in protein synthesis. So this portion, you've gotten kind of an overview of the history of the discovery of DNA and its relationship to genetics and its relationship to carrying certain physical characteristics from one being to another. We'll pick up on topic 2 in just a moment.